Continue our recorded session on cell cycle. So we're now at the mitotic phase or the M phase, the cell division proper. So the M phase represents the phase when the actual cell division or mitotic division happens. No? So it starts with a nuclear division. So the nuclear membrane disintegrates. No, and that corresponds to the separation of the daughter chromosomes, the daughter DNA, into two, and will end with the division of the cytoplasm. Okay. So there are four steps. We have the prophase, metaphase, anaphase, and telophase. And later, when all of these stages have when all these stages already transpired, that we expect the cytoplasm of the dividing cell to be distributed into two daughter cells. So just as a review, let's look at chromosomes. No, so in order for mitotic division to be successful, okay, chromosomes must be duplicated in number and must be physically separated during mitosis. So mitosis facilitates that physical separation. Um, Looking at the structure of chromosomes, so chromosomes are tightly packed DNA. Okay, primarily they are I think I've already discussed in the lecture that DNA strands are very long, so they have to be condensed in order for them to occupy, or in order for them to be uh, properly localized in the nucleus. So we represent the chromosomes with figures that look like the letter X, right? That after DNA replication, okay, you can expect them to appear like this. Let's have the laser. So single strand will later on become two strands after DNA is replicated or synthesized. Right. So the structure of the chromosome, a general overview. Okay. So there are two identical chromatids that are present after the DNA is synthesized, the DNA is replicated. The short arm of chromosomes is called the P arm. No? And there is a constriction between the chromatids called the centromere. No? So it's a constricted point in the chromosome that sort of divides, or sort of will be what do you call this, a basis for the separation of of the DNA, of the duplicated DNA material later on. No, the long arm is called Q arm. So short arm, long arm, and the centromere. Okay, now let's take a look at the stages of mitotic cycle, or the mitotic division. So let's have P for prophase. So prophase is characterized by four different events. So first, chromosome condenses. Okay. Then second, the nuclear membrane disintegrates or disappears. Third, centrioles, which are important organizing centers for fibers necessary to cytoskeletal fibers necessary for division. They separate, and they will be found on opposite poles of the dividing cell. Then spindle fibers form um, and radiate toward the center of the cell. So here you can see in illustration, the centrioles are beginning to move away from one another and be positioned on the opposite poles of the cell. Next, the metaphase. Metaphase is characterized by two events. Chromosomes line up across the middle of the cell. So they line up across the what we call the metaphase plate, right? And the spindle fibers radiating from the centrioles, they connect to the centromere of each sister chromatid. So the connections here represented by the gray lines, okay? So these are spindle fibers radiating from the centriole, connecting with the centromere of the chromosomes. No? Then anaphase is characterized by three events. First, centromeres that join the sister chromatids will split, right? Then sister chromatids will separate. They will become individual chromosomes later on in each of the two progeny cells. And separated chromatids will move to the opposite poles of the cell. So usually it looks like this illustration here. Then we have telophase, characterized by four events. Now that the chromatids have been separated already to two progeny cells, chromosomes will begin to uncoil. The nuclear envelope, the nuclear membrane will reform around the chromosomes. Then spindle fibers will break down. They will dissolve because they're no longer needed. And cytokinesis will begin. So the cytoplasm distribution to two cells will now commence. Then cytokinesis is the final division of the cytoplasm into two individual cells. So in animal cells, the cell membrane will form what we call a cleavage furrow that will eventually pinch the cell into two equal parts, almost equal parts, no? Each containing nucleus, organelles, and of course their copy or its copy of the replicated DNA. So just as a summary, you have a very good figure here. So please take note of the key things that are happening or the how the structures, organelles, associated organelles are involved from prophase until the cell produces two daughter cells. Okay? Another type of cell division is meiosis, which happens in sexually reproducing or happens during or in cells that are involved in sexual reproduction, the sex cells, the gametogenic cells, the egg and the sperm. No? Unlike mitosis, in meiosis, okay, there is what we call crossing over. Okay, so crossing over is identified or is defined as the mixture of chromatid sections such that genetic variation occurs. No? And depending on the type of the gametogenic cell that is dividing, no, um, the number of the daughter cells is not always uh, two. No? Like uh, um, for the case of I think for humans, no, the meiotic cycle will produce haploid daughter cells with uh, very different character, with very different genetic makeup from one another because of crossing over. Yeah, so here is how crossing over happens, right? So the chromosomes, okay, from the homolog pairs, okay, per chromosome number or chromosome set, okay, they associate the long arms associate the chromosomal arms associate with one another. They will carry out genetic material exchange in this position, allowing for variation in the genetic makeup of the daughter cells. Okay. So here we can see um, I think this is human, the karyotype of the uh, human cell, human bodily cell. Okay. What else? So we mentioned I've already talked about this chromosome. Yeah. So just to I think one thing I have to mention is that the crossing over happens between non-sister chromatids. No. Okay. So this is more on the process. I don't think I have to that we have to be introduced to these stages or to these processes yet, because I think in your genetics class you will be discussing more about genetic variation arising from meiotic processes in gametogenic cells. But yeah, as you can see, the crossing over process is a highly complicated one. Will require rearrangements, no? Physical rearrangements of genetic material in the involved chromatids. Okay. Now, some key differences. I mentioned, I think the first check itemed, and then mitosis ends with two cells. Meiosis will end with four cells. Diploid in number for mitosis, still the number of chromosomes as the parent, while for meiosis, the daughter cells will have half of the number of the chromosomes of the parent cell. And in terms of genetic identity, the daughter cells are genetically similar with the parent cells, while for meiosis, the daughter cells are genetically unique. They do not have the same genetic makeup as the parents and as with one another. Okay? Then some similarities. Okay, they start, of course, both processes start with one cell. Both undergo or require both undergo nuclear cytoplasmic division. Both will involve disappearance of the nucleus and the nucleolus, as well as disintegration of the nuclear membrane and its reformation later on. Both involve formation of spindle fibers. Both will, of course, transfer DNA from parent to progeny, and both are types of replication and reproduction in the sense of producing, in the sense that both processes will lead to formation of new living cells. Okay. Now, for our model systems, for mitotic division, we'll be making use of the whitefish blastula, the whitefish dividing or a specimen representing the blastula of a whitefish. No? So blastula meaning it's a ball of cells after an egg has been fertilized. So an egg that has been fertilized will of course will become a zygote, and that zygote will continue on dividing, continue on doing cell division. So you will be able to see or observe different stages of the mitotic cycle and the preparatory stages also in our whitefish blastula model system. Okay. So you will identify or you will look for cells undergoing the different stages of mitotic cell division. So how you can scan, what you will see in your laboratory proper or your journey lab proper is specimens, prepared specimens. So you would have to scan the entire section, the whole section, for you to be able to see the dividing cells. No? And you can follow the direction that was shown. Okay, so that ends our pre-lab discussion for our module on cell division.